All right, we're moving through this dot point now. So last week we looked at the sources and mode of transmission of the various signaling molecules. So the hormones, neurotransmitters, cytokines, and pheromones. And today we're going to look at these two sections here. It might look like a lot, but they all relate. These two sections relate to each other really well. So we're looking at the stimulus response model and this kind of term called signal transduction, which is a really important term that you need to learn. And then we're also looking at how this signal transduction is different depending on whether we have a hydrophilic or a hydrophobic signaling molecule. So just to recap what we looked at last week, so let's say we've got cell number 1 here and cell number 2. Okay, and we're looking at signaling molecules. So this cell here produces a type of signaling molecule. Could be a hormone, it could be a pheromone, it could be a cytokine. But it produces a signaling molecule, and then that signaling molecule travels and binds with a receptor. Okay, so binds with a receptor. In this example, it's going from one cell to the cell next to it. So this would be an example of a paracrine signal. But remember that this could also happen in the same cell, so it could all stay in cell number 1 where it would be autocrine, or it could be traveling to a completely different area of the organism and it could be endocrine. But I just wanted to recap that because what we're looking at is kind of how this happens, what happens once a cell receives. In your notes, so we're still in the section of cellular signals. You need to make a heading called the stimulus response model. Now, most of the information I'll put in these slides, you should be taking notes on, unless I specifically say you don't need to have those notes. This is what I would normally be doing in class on the board. Okay, so stimulus response model heading. And this model describes how a system responds to a stimulus. And this might ring a bell from something we looked at last year and you might have looked at in the past when you've looked at things like the nervous system. But we're looking at it at a cell level now, and we call this signal transduction. So you need to have this written down. So signal transduction is a 3-step process. The first step is reception. Okay, and that is when a signal, so the signaling molecule, is received, so reception, received by a receptor. Okay, and you could even add in here a specific receptor, because remember this receptor has to be specific to the signal. So step 1, reception. Step 2 is called transduction. Now, transduction is a series of events that occur in or between cells. Most of the examples we will look at is inside the cell. Okay, we just have that written under that. So we've got the signal received in reception, a series of events occur in the cell, and that is known as transduction, and then we get our response. And so this is when a change is carried out by an effector. So that word, if you think about it, effect, you kind of have cause and effect. So change is carried out by an effector. In the past, you would have looked at this in terms of a stimulus and a response, like the heading is called. but at the cell level, we refer to it as signal transduction. So reception, number one, the signal is received. Number two, a series of events happen. And we'll look at some of those, but some of them you don't need to know in a lot of detail. And then number three, we get the response. They have to go in that order. You can't have the response without having the first two things happen. I just want to show a little example of this because I think it might help to understand what we're talking about. So we're going to look at, excuse me, at insulin. So insulin, if you remember from last year, is involved in decreasing blood glucose levels. And how it actually does that is by getting cells to take in the glucose. So if the cells take in the glucose, it's not in the blood anymore. And this is a nice example of signal transduction. So reception, number one, we have insulin. So that's our signaling molecule, binds with the receptor. Okay, and that is an insulin receptor, specific receptor. So over here, this is our insulin binding with the receptor of a cell. I'm just going to point out as well that these blue bits here are the glucose. So glucose is outside the cell, and we want that glucose to come into the cell. Transduction. You don't need to know the details of all the little things that happen, but let's just say we have a series of events in the cell. Okay, so I'm going to show that as being these little red things. So after the signal is received, other messages get sent. Okay, that's all you need to know for that. Transduction, number two, a few things happen in the cell. And number three, we get our response. Now remember, the aim of this is to get glucose into the cell. So the response here is that the channel proteins that are in the membrane open and glucose enters the cell. Okay, so down here you can see that that channel has opened up and now that glucose is moving into the cell. Okay, so if you want to draw something like this in your notes, or there'd be a lot of pictures on on Google, or you could take the ones from my slides. But it just shows you we've got the message getting received over here. So signal transduction with signal reception with insulin binding to the receptor. We have a series of events happening in the cell. That is our transduction. And we have a response. The channels open up and glucose enters the cell. All right, I just want you to make a little summary of what we're going to go through in your notes now. So there are two pathways of signal transduction. So two ways basically that the signal can be received. The first pathway is the intracellular pathway. And this is when the signal binds with the receptor that is inside the cell. So let's say, for example, I've got a signal. So this molecule is traveling and it goes through the cell and it binds with a receptor inside the cell. Okay, so that is an intracellular pathway. The second pathway is a membrane pathway. And this is when the signal binds with the receptor that is embedded in the membrane. So let's say we've got a signal traveling from a different cell, and instead of going into the cell, it binds with the receptor on the outside of the cell. You can draw something like this as a nice simple summary. Otherwise, I'll have some more detailed pictures as we go through them. For the first one, the receptor is in the cell. The second one, the receptor is in the membrane on the outside of the cell. We'll start with the intracellular pathway. This all comes down to whether or not a molecule So the signal is hydrophobic or hydrophilic. So it's really important that you take these notes down. So hydrophobic molecules, which remember are non-polar molecules, can diffuse across the plasma membrane. So they are, remember, they are lipid soluble. So they like fat, so they can travel through the fatty acid tails of the plasma membrane. So a hydrophobic molecule, which you can see here, can go through the plasma membrane. And once it's in the cell, it can bind to a specific receptor inside the cell. It's still, keep in mind, it has to be specific still. It has to be the right shaped receptor for that molecule. Okay, so it's gone in, and you can see here that the receptor and the signal, in this example, it's a hormone, have bound together. And we call this a molecule receptor complex. In this example, it says hormone receptor complex because it's specifically talking about a hormone. Okay, so it makes a complex when they're bound together, similar to, remember how our enzymes and substrates make a complex? These make a complex bound together. And then that complex will carry out or initiate the response. So if we look at this, we could say we have reception happening in number 1. We've got a couple of things, so between 2 and 3, we've got transduction. So there'll be a few events that happen that we don't need to know the details of, and then we're going to have our response. It is important that you know a few examples of what a response might be. What might a cell do as a result of a signal that it has received? So there's 2 things it might do in terms of gene transcription. So a signal may initiate or inhibit gene transcription. So it might turn on or turn off a gene. And in this example, it's turning on the gene because we're going from DNA into mRNA. So it's transcribed, and then that will end up going through to translate a protein. Another thing that a response might be would be to activate or repress enzymes. Okay, so any of these things will have effects on cell functioning. It might be turning something on or turning something off. And some examples of molecules that are hydrophobic and will go into a cell are steroid-based hormones. So steroid hormones are lipid-based. Okay, so testosterone and estrogen are examples of that. They can go into their target cell and carry out their response. The next pathway is the membrane pathway, when we have membrane receptors. So examples of molecules that use membrane receptors are all of our protein-based hormones, as well as neurotransmitters. They always bind to a receptor on the outside, and cytokines as well, which we'll look at in a week or 2. So as you might have guessed, hydrophobic molecules can go in. Hydrophilic molecules cannot enter the plasma membrane. They are polar. Okay, so they can't get through here. So there's no point having a receptor inside the cell if the molecule can't get in there. So they will bind to a specific transmembrane receptor. Remember, we looked at the plasma membrane. There are different types of proteins. So we have this receptor here, this transmembrane receptor, and you can see it goes the whole way through the plasma membrane. That is quite important. It has an outer bit on the outside of the cell here, and then it has an inner part. So the signal binds to the receptor. And then remember that receptors are proteins, so their shape is so important. When this happens, when this signal binds with the receptor here, that will change the shape of the protein a little bit. It'll change the shape of the receptor. And what happens is on the inside now of the cell, it's the same receptor, that will change shape. And when that happens, it activates what are called secondary messengers. Okay, so secondary messengers are other molecules that are inside the cell, and when this kind of chain of events happens, the receptor binds here, changes the shape of the receptor, and then these molecules inside get activated or released, and they're called secondary messengers. You don't need to know too many specifics of that, but a term that comes up a little bit is called a G protein, and that is an example of a specific type of secondary messenger. So I'll put that there because it can come up in examples used. And then this kind of series of events that happens is called a transduction cascade. Now, if you think of a waterfall that cascades, you've got lots of things moving, it's kind of based on that, because we had our first messenger up here, our transmembrane, our initial signal activated these messengers inside, and then they will go on to activate other messengers. So it's kind of like a chain, and just it's worth having that word cascade written down because it does come up a little bit. One thing I'll add here, this word ligand, which you may see come up, basically is just a small molecule that binds to a larger molecule. Okay, so it's a general term. It's not in the study design specifically, but it's worth having it written down in case you see it in examples. So small molecule, for example, our signaling molecule that binds to the receptor. And once all of these events have occurred and we've had our cascade of secondary messengers basically passing on that message through the cell, we get our cellular response. Okay, so cellular responses here. So it is a bit more complex than the other example because there are more things involved. Another term that you should know is signal amplification. Now, if you think of what happens when you amplify music, you're kind of making it louder. You know, if you've got a speaker, you see a picture of, you know, how music would be amplified, it kind of goes out like that. And you should think of that when you're thinking of this example. We're basically starting with one signal, our primary messenger, and that activates a bunch of different messengers, and that is called signal amplification, when several secondary messengers are activated at once. Now, just listen carefully. I do not want you to look at this picture over here and think, oh, I don't know what any of those words are. You don't need to know this specific example. You need to know the term amplification, but you don't need to know this camp example. But if we look at this picture, this would have been our original receptor and our signaling molecule. So it must be hydrophilic, can't enter the cell. This is our transmembrane receptor here. So it's going the whole way through the cell. Once this message is received, it switches on a bunch of different things at once. And that allows for the message to be amplified. Okay, so that's all you need to know about amplification. This is just another diagram that shows the same thing. So we have our signaling molecule. It binds with a transmembrane receptor. So that tells us that this molecule must be hydrophilic. If it was hydrophobic, it would just go straight into the cell. So it binds here, reception. It turns on these molecules. So these are our secondary messengers. In this diagram, they're called relay molecules. They're the same thing. They're relaying a message. They're our secondary messengers. This is the term that VCAR want you to use, secondary messengers. So that's our process of transduction, and then eventually we'll get our cell response. So remember, that could be something like switching on a gene, or it could be activating an enzyme. Okay, they're examples of responses. I'm just going to show you a video now. It's a really good animation of how this occurs, but I'm just going to warn you that the volume's a little bit loud on it. I'll try to be able to speak with it, but my voice will be quite quiet on it. The other fun thing about this video is that it's in German. There's no talking in it, but the little subtitles are in German, so you can learn some German. Okay, so we've got this signaling molecule here, signal molecule, okay, and that's the cell membrane there, and you can see lots of receptors on the cell membrane, because there would be lots of different receptors. Now, the molecule can't bind to that receptor because it's not specific. It has to bind to the specific receptor, which it does. Okay? Now, inside the cell, protein change shape and other proteins are activated. Don't worry about the names of them, but like I said, the G protein is an example of other molecules inside the cell that now start to carry out different functions. And don't worry about these names, more just to see what actually occurs. Okay, so other things that are being activated. Now this would be an example of some signal amplification. Lots of things are getting switched on at once. Okay, and I'm going to leave it there because the rest of it will be complex. Okay, that was fun. All right, this is an exam question from a few years ago. A signal transduction pathway is shown. Some of the parts of the pathway are labeled. So A, M represents a compound which could initiate signal transduction. Give an example of compound M. Okay, so this could be basically any type of signaling molecule that we know binds with a receptor on the membrane. So we just talked about a few then. We talked about protein-based hormones. Okay, so for example, insulin, like the example I used at the beginning, it could be a cytokine. Okay, so that just asks for a general example. And then question B, the initiation of signal transduction is controlled and specific. Explain what features or what feature ensures this specificity. So basically, what makes the switching on of a series of events specific? And hopefully you know that that is the fact that the receptor is complementary in shape to the signaling molecule. So exactly the same as when we looked at things like the enzyme substrate complex. They have to be the same shape for it to be specific. Okay. Now, this question doesn't ask it, but you could probably figure out a lot more things from this picture. It's showing that our receptor is found in the membrane. It's showing that we have these secondary messengers being part of the process. So we know that this is a membrane response as opposed to an intracellular response. All right, I'm going to get you to pause here and have a go at this. This might be a really good picture to take from my slides and to maybe put into your notes if you have access to a printer or you're doing online notes, or even drawing something like this, because it summarizes these two things quite well and you could add in the notes. So pause this and then have a go. Okay, so it's asking you whether hormone A and B, whether they are hydrophobic or hydrophilic. So let's start with hormone A. We can see with hormone A, the receptor is inside the cell. So therefore, it is hydrophobic. So it is the lipid-based hormone. And the reason for that is because it is able to bind with a receptor inside the cell. So you would need to make a bit of reference there. It's able to bind with a receptor in the cell, so it must be hydrophobic. Okay. B would be our protein-based. And if we look at the picture with B, it has bound to a receptor in the membrane. So it must bind with a receptor on the membrane, so it must be hydrophilic. Okay, if it was hydrophobic, it could pass into the cell. Okay, what does C represent? So C over here, so it's on the side of our hydrophilic signal where it bound with the receptor on the membrane. So C would be our secondary messenger. Okay, so it's the messenger that is signaled or switched on by that initial reception. And the function of C would be to relay the signal to the effector. Okay, so it's basically that gap between the reception and the response. A couple of challenge questions there. Describe the process of transduction. So you would need to actually explain each of these. So a signal is received by a receptor. A series of events occur inside the cell, and then the response is carried out by the effector. And the final bit here, give 3 examples of cellular responses that might occur. So you could say gene expression. You could say, remember that insulin example, that channels open to allow molecules in. You could say enzymes are activated. Okay, all examples of responses that might occur as a result of signal reception. Okay, I have put some other things that you need to do in terms of Ed Rollo with this. Now I know this is probably more than a single session's worth of work, so I will make sure that our next apoptosis lesson for Tuesday afternoon is probably a bit less than a single's work. So between this and that session, it should kind of even out. All right, I'll chat to you soon.